about the functions. We'll go a little bit endocrine on you, okay? We're going to talk about CCK, cholecystokinin, all right? And I talked about a little bit when we were discussing the gastric phase of digestion about, you know, how the stomach motility increases with the presence of food and the secretions in the stomach increase. And then when we transition into the intestinal phase, all right, the stomach secretions decrease, the stomach and the motility decreases. Part of that is through the nervous innervation, okay, from the autonomic nervous system. But also, there's an endocrine component to that, and that's because the small intestine will secrete or release cholecystokinin, CCK, and secretin, which will decrease the stomach's motility and secretions. So that's when we first heard about it. But really what we're going to see CCK plays a big role in is when the chyme, all right, gets dumped into the duodenum and it has a high fat content, okay, it's going to stimulate the gallbladder to contract. Okay, the muscularis layer of the gallbladder will contract, all right, it'll secrete the bile out of the gallbladder, and with the liver too is always producing, not always, but it's producing bile too, and so that bile will then enter into the small intestine along with, all right, our pancreas, because at the same time, while we're stimulating the gallbladder to contract and release its bile, the pancreas is going to be secreting its digestive or pancreatic juices, all right, at the same time into the small intestine. So we're going to see that chyme get mixed up with a lot of things. Now, we'll talk about the functioning of the bile salts in a little bit, but its job is the process of emulsification, okay, which is basically breaking down bigger fat molecules into smaller fat molecules, or big small fat or lipid droplets into small lipid droplets. So that's what the bile will help to do with that. And then the pancreatic juices here, all right, we saw, and if you don't remember, all right, the ingredients here for our pancreatic juices, all right, a lot of water, all right, bicarbonate, and then we're going to have, all right, enzymes that are going to help to digest starch, all right, more fats, okay, and we're going to release, this is important, the inactive protein enzymes, because they can't be active. We saw that in the stomach. Remember pepsinogen, right? The cell can't have an active protein enzyme inside of it. There's proteins all over the place. It'll start degrading all the proteins inside. So it's going to secrete inactive proteases, okay, which will then be activated, and we'll talk about that. All right. So a big component of our pancreatic juice is going to be, yes, water, right? Bicarbonate, that's what that is. Okay, that's going to help to neutralize the acidity of the chyme, all right, and then our digestive enzymes here, all right, and not to forget also, I didn't mention this, but the nucleases, that's going to be, all right, the enzymes that are going to help to break down nucleic acids, okay, DNA, RNA, all right, blah, 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 okay, so the other not enzyme, but a hormone that's going to be released is going to be secretin. And we saw before, all right, secretin will also inhibit gastric secretions and motility, all right, but it's going to play a huge role, all right, in the release of a basic solution in the small intestine. Again, this will help to neutralize the acidity of the chyme. All right, so we got a lot of stuff pouring into the small intestine, into the duodenum here, all right? And if you think about it, a lot of it makes sense, right? We need to, one, neutralize the acidity of that low pH that's coming in from the stomach, okay? So we're going to start to do that because mainly hydrochloric acid, all right, its job is to help to denature proteins, okay? Helps to break down, because proteins can be, at a minimum, 200 amino acids long. It can be much longer than that, 300, 400, it can be quite big, okay? But at a minimum, for a structure to be considered a protein, it has to have at least 200 amino acids, right? So we'll talk about the other classifications, all right, of the sizes of some of these structures. So you'll hear of oligopeptides, and the legal peptides are anywhere from 3 to 20 amino acids long. All right, so keep in mind, all right, when we're dealing with our duodenum, all right, right away, we're going to see the secretion, all right, of two hormones, secretin and CCK, all right, in response to that chyme entering into the duodenum there. All right, so let's move on into the large intestine, okay? So we remember there's three parts to the small intestine: duodenum, jejunum, and the ileum. So the ileum, all right, is going to attach itself, all right, onto the cecum, okay, and dump that chyme into the cecum, where we're going to continue on with the digestive processes of water absorption, nutrient absorption, right? But think of the large intestine as where we're going to start to form our feces, okay? We're going to still get absorption. It still occurs there in the large intestine, but now we're going to start to form our waste here, all right? So we're going to see the majority of the large intestine here in the abdominal region here, all right? Remember, it forms that cecal sac there, and it goes up to the ascending colon and kind of makes an inverted U here, all right? Then comes back down, then goes down into the medial portion of the pelvis and forms the rectum, okay? We're going to still see a lot of absorption here, but not only that, we're going to continue to have absorption of water. The majority of the water gets absorbed in the small intestine, anywhere, I believe, depending on what your water intake is, anywhere from 8 to 9 liters a day. It's a pretty decent amount, okay? We're going to still absorb the electrolytes here, right? But now that chyme is going to be converted more into the feces, which is going to be less watery and more of a solid, okay? And then eventually, all right, we will prepare the feces for elimination, okay? Store it in the rectal area, all right, until there's enough that's built up there, until it's time to eliminate the waste through the anus there, okay? So let's start off talking about the parts of this large intestine, all right? That first kind of like sac-like structure, all right? That's the cecum, okay? It's in the right lower quadrant, all right? Also, we'll see the vermiform appendix that comes off inferiorly off the cecum. All right, you will house in the vermiform appendix, all right, bacteria. It's believed to be what we call beneficial bacteria, all right, but it's loaded, all right, with secondary lymphatic structures. All right, so you can see how, one, you've got secondary lymphatic structures combined with bacteria. You can see that it has a potential recipe for disaster. All right, because if something occurs with that bacteria and it turns into what we call the opportunistic, all right, organism, then we can get appendicitis, and that's a problem, okay? So both of these structures are going to be intraperitoneal, all right? After this, then we're going to start alternating parts of the large intestine from intraperitoneal to retroperitoneal. So meaning, right, the cecum and the vermiform appendix are both intraperitoneal. The next part of our large intestine is the ascending colon. All right, that's going to ascend on the right side up towards the liver. So that part is going to be retroperitoneal, okay? And so it extends upwards towards the liver, where it's going to take that 90-degree turn, all right, in which we know it as the hepatic flexure or the right colic flexure. Either or is appropriate, all right? It's just we call it the hepatic flexure because that's where the liver is, all right? So this ascends up towards the liver. It takes that 90-degree turn, okay? And then it's going to traverse from the right side of your body over to the left side of the body. Right? At that point, all right, we are now in the transverse colon, okay? That is intraperitoneal. All right, and it crosses over the abdominal area from right to left until then it gets to the side where the spleen is located. And then it takes a 90-degree turn inferiorly, all right, and heads down towards your left lower quadrant, all right? And so we call that turn there the splenic flexure or the left colic flexure. And then that part of the colon that descends down to the left lower quadrant is our descending colon. Once again, we have a retroperitoneal because we're alternating, okay? And then this structure, the descending colon, all right, will take kind of like a quick turn, and it'll start to head medially and inferiorly down towards the center of your pelvis. Okay? So it comes down on the left side of the body, and then it takes this kind of S-shaped configuration and goes down into the medial portion of your pelvis. So that S-shaped configuration of the colon is easy to remember because it also starts with an S, and that's the sigmoid colon. Again, this is another intraperitoneal structure. Okay? It's going to wind up in the pelvic cavity where it'll terminate as the rectum. Think of the rectum as the storage facility, all right, for feces, okay, for your digestive wastes, okay? And here at the rectum, again, we have another structure that is retroperitoneal, all right? And this is where we're going to store our feces, and when we build up enough of it, we'll excrete the feces out through the anus. Inside here of the rectum, when we talked about this in lab, we have these transverse folds here called the rectal valves. Okay? So that'll give kind of almost like a segmented configuration to the feces during the excretion process there. Okay, so it is a storage site, all right, and it is prone to distension. And if you're not familiar with distension, that is just like a stretch of the wall there, all right? And so when we start to increase the amount of feces there, it'll distend the rectum. The baroreceptors in the rectum will transmit sensory information, all right, to your central nervous system. Okay, and this is where you're going to start to feel the urge to defecate, to use the lavatory. Okay, so we have smooth muscle in that rectal wall there that will help with expelling the feces. We're going to actually get into the defecation reflex here shortly, all right, but not right now. So here we can see, all right, on our large intestine, here's the ileum coming in and terminating onto the cecum here and the ascending colon where we have the ileocecal valve here. All right, so this bottom sac here, that's the cecum with the vermiform appendix hanging off. Ascending colon, retroperitoneal. Transverse colon, intraperitoneal. Descending colon, again, retroperitoneal. Sigmoid colon coming in intraperitoneal. And then we have our rectum, another retroperitoneal structure where we have the formation of the feces here. Just waiting for the expulsion, all right, or elimination through the anal canal here. All right, speaking of the anal canal, all right, it is lined. It's important that you know this by stratified squamous epithelium for mechanical stress, all right, for the actual elimination process of the feces here, all right, and this is where we're going to see, all right, the anal canal will literally travel through the levator ani muscles. If you're not familiar with what those muscles are, those are the muscles that make up your pelvic floor. I've said this before, I think, to you guys. That is the muscle group that literally holds your guts into your pelvis. Okay, so everything that sits there in the pelvis, all right, will be held in, all right, through the levator ani muscles, and the levator ani muscles which will also help with the defecation reflex here. Okay, so we have in the anal canal. Come right back to this. I just want to show you what I'm talking about. Okay, all right, you've got your the renal columns here. All right, and we also will have, all right, the fissures in between here, the sinuses. Okay, so you see these raised little columns here, and in the sinuses here, that's where, again, like I said, you can never have too much mucus. All right, so you have our glands here that are going to produce mucus to help ease the passage of the feces through the defecation reflex here. So there's two valves that are involved here, all right, or sphincters that are involved in the anus. You have the internal anal sphincter, all right, which is part of, all right, it's smooth muscle, so we know if it's smooth muscle, it's involuntary. Okay, it's like an extension, all right, of the internal rectal muscular wall there. Okay, so it's smooth muscle, So that muscle will be controlled by the autonomic nervous system. All right. Then we have the external anal sphincter. All right. That is going to be voluntary, so it is going to be skeletal muscle, which you can control by the somatic nervous system. All right. So when you are actually trying to, all right, get rid of the feces, the external anal sphincter, this is the sphincter that you learn to control during that toilet training or potty training, whatever your family calls it, during that process, all right, when you're a child, okay? So both of these valves are usually going to be closed except during that period of time when you're going to be defecating here. All right, so let me kind of show you a quick picture here, all right? All right, getting down here into the rectum, you see the rectum has this muscular wall all around it, all right? And as we get down towards the anus, all right, it's still smooth muscle, right? Then it becomes the internal anal sphincter, all right? Then you can see here we got the levator ani muscle, all right? That's still skeletal muscle. And down here by the exterior portion of the anus, all right, that part becomes the external anal sphincter. That is under voluntary control here, okay? If you're ever wondering about hemorrhoids, here you go, the rectal veins right there, okay, when they become damaged. All right, and it's very helpful too because if people are passing blood in their stool, has anyone ever heard of a stool guaiac test? It's basically a test which determines if there's blood in your stool, okay? And sometimes you can tell, or it's helpful when you see the color of the stools. If it's bright red, then it's fresh blood, and so it's closer to the actual distal end of the digestive system. Right? If it's black and tarry, all right, you know what happens to the blood? It coagulates and it becomes darker over time. Okay? So if it's dark, all right, tarry stools, then you can realize, okay, then this the bleeding is going to be further up in the digestive tract. Okay, so these are just different ways to kind of help determine what the heck is going on. All right, so if you notice on our large intestine here, and we talked about it before, all right, you had to label that. You'll see here in the large intestine, all right, these strips of smooth muscle, and they're longitudinal smooth muscle, okay? So we call that the tinea coli. And it's these tinea coli, all right, that are going to help to form those sac-like structures called haustra. Right? And because it's the longitudinal muscle, we know that the longitudinal muscle helps with movement of digested food, whatever, through the GI tract. Okay? So it's the tinea coli, and we'll talk about this in a moment when we talk about movement here in the large intestine. But the tinea coli are going to help to advance, all right, that food through your GI tract here. Okay? So we like to refer to it as like a waistband here. All right, and then on the outside, you'll also have these globs of fat that hang off the external portion of the large intestine, and those are called omental appendices. All right, it's just accumulations or these lobules of fat that you'll see just kind of scattered throughout the large intestine there. All right. All right, so you heard me talk about bacteria, all right, that hangs out in, all right, the appendix there, okay? But you actually have a natural flora of bacteria in your body, okay, which actually helps you to digest, all right, food. All right. Now, a lot of the times when you're born, at least in the United States, all right, they'll give you a vitamin K shot, okay, because you haven't had vitamin K production because you don't have this normal flora of bacteria. So we give it a name, indigenous microbiotica, all right, and it's this normal, we call it normal, I mean, how normal is bacteria, all right, but it's the normal bacteria flora, which means it lives, all right, symbiotically with you, all right, as long as you don't bother it, it won't bother you kind of thing, all right, but it helps with the digestion, the further breakdown of, look at that, carbohydrates, proteins, and lipids. It helps to further break that down, but it also will help to produce vitamin B and vitamin K. Big deal, vitamin K. Vitamin K is needed for clotting, okay? That's why you get a shot when you're an infant, because you're not producing the vitamin K. You still need to be able to clot. This bacteria helps with that, all right? So we'll see, all right, over time, this bacteria will start to develop, and it becomes part of the normal flora. Now, if you have any type of perforation to the large intestine, then that's when that bacteria can become a problem. Okay, if it gets out of this environment, all right, then you can start to worry. That's why it's one of the things that they're worried about with gunshot wounds, stab wounds, any type of puncture wound that you may sustain to your abdomen. They worry about bacterial infections, both from the external environment, but also from the internal environment. Okay? All right. Feces, all right. Yes, it's the final product of the large intestine. It's primarily made up of water and salt, epithelial cells, because remember, as those feces and that digestive food product is rolling through your GI tract, it's going to cause friction. It's going to scrape and damage epithelial tissues, all right? And it gets picked up in the feces here for the eventual, all right, elimination. We're also going to see undigested material like fiber, cellulose, okay? And of course, some of the bacteria that lives happily in your digestive system, it gets caught up too, and it'll be excreted along with everything else that's in the digestive tract there. All right, so movement of the food product through your body. There's two parts, just like that. Keep in mind, when we're moving, all right, this digestive material through the small intestine, through the stomach, all right, through the large intestine, there's two types of movement. There's going to be the mixing movement, all right, what we call the haustral churning, all right. So this is going to be again, we're going to continually try to mix this up and expose as much of it to the intestinal wall as possible to maximize absorption, to get that stuff through. So this haustral churning, all right, is going to cause, all right, think of it more like a local reflex in which we're just going to move, all right, that digested material, the chyme there, from one haustra to the next one over. That's all we're doing, all right? So we're going to move that material to the more distal haustra. All right, so that's kind of local. Then we have this other phenomenon called the mass movements. This here now is when we're really going to see a significant movement of that chyme through the the GI tract. So hence we call it mass movements here. All right, you have this occurs a couple times a day. And what stimulates the mass movement is when food enters into your stomach. Okay, because like I said, when you're starting to bring food into your body, all right, your GI tract is going to make room for it. All right, so when food is entering into the digestive tract and it enters into the stomach, all right, your stomach is going to cause, we're going to see it here in a moment, what's called the gastrocolic reflex, which is going to stimulate a mass movement to really move not just, all right, food or the chyme from one haustra to the adjacent one. It's going to move it several haustra down, all right? Always moving that digested material, all right, towards the rectum. So think of the haustral churning as a small one, and the mass movements, all right, are going to be huge, all right? And the Taenia coli are going to play a huge role in that, all right? So these mass movements are from what we call the gastrocolic reflex. We're going to talk about that in a moment. We already talked about the gastroiliac reflex, all right? When food enters into the stomach, it stimulates the ileocecal valve to open up and release chyme into the cecum. Again, trying to get things ready for that food that's in the stomach, because remember, the stomach is a holding bag. It's not going to hold on to that food forever, just temporarily, all right, before it moves it into the small intestine. So that's what we're going to see here with these mass movements. They're a way, we're just going to clear the pathway, get stuff out of there. Let's move some of this food material that we just ingested into the small intestine. Let's move what's in the small intestine into the large intestine. All right. So the gastrocolic reflex, all right, will initiate or cause the mass movement here. All right. Then, of course, we'll see the defecation reflex. Kind of seeing the pattern here. A lot of this GI tract is just reflex based. All right. Some sort of stimulus occurs. Then we get sensory input from that stimulus, which results in some sort of motor output effector, all right, type of response. Okay, remember, the effector organs that we're going to be dealing with here are going to be mainly smooth muscle, which is going to cause mixing of the food product and advancement of the food. All right, and then we'll also get glandular secretion. All right, so we're going to get those hormones being secreted and some of those digestive enzymes as a result of that. okay? So the defecation reflex here, and I'm going to go over this with you here in a nice picture, all right? I'll set the table for you, okay? So you just had a big meal, hour passes, you start to get the urge to go to the bathroom. That is because, all right, feces have moved into the rectum. It's causing the rectal to start to become somewhat distended, puts a stretch on the wall of the rectum. All right, those baroreceptors monitor that. They send their sensory input information to the spinal cord. This is a spinal reflex. I don't know if you remember reflexes very well from 210. All right, there's several different types of reflexes. A reflex is either going to be cranial or spinal. If it's cranial, that means all that information goes up to the brain, and the brain somehow is going to affect the response. This is a spinal reflex. It doesn't even go to the brain. Okay? It just goes to the spinal cord. Okay? So it's going to go to the spinal cord. All right, that sensory neuron is going to connect to a motor neuron and stimulate the walls of the rectum to start to contract. They're smooth muscles. So that's innervated by the autonomic nervous system. Okay? It's going to cause those muscles to start to contract, and it's going to cause the internal anal sphincter to start to relax. All right? Still got a problem, though. We got to deal with the external anal sphincter. No problem. This is where the somatic nervous system comes into play. The somatic nervous system, your cerebral cortex, the sensations of having to go to the bathroom will reach your consciousness. Like, hey, you know what? I got to go to the bathroom. All right? So as that's occurring, all right, well, hopefully it's not as it's occurring. You're sitting down while you're doing this, all right? But when it's time to actually have a bowel movement, all right, the somatic nervous system will then start to trigger or affect, all right, the skeletal muscle of the external anal sphincter. You cause that to relax, in which you can actually have a bowel movement. At the same time, while you're trying to, all right, move the feces out of you, you're going to be utilizing what we call the Valsalva maneuver. Is anyone familiar with the Valsalva maneuver? Basically what it is, and I mentioned it when we were talking about the respiratory system, it's an increasing intra-abdominal pressure against a closed glottis. So it's like asking somebody to take a deep breath in and bear down like you're going to have a bowel movement. Obviously don't have a bowel movement, but don't exhale, okay? So you're going to increase the intra-abdominal pressure. That's going to help, all right, to move the feces out of the body. Okay, so here's your picture. Basically what I just talked about. All right, you have the baroreceptors here in the rectum. As it's starting to fill with the feces, it's going to cause a stretch. That sensory information is going to travel here to the spinal cord. Okay, where then it'll directly synapse onto a parasympathetic motor neuron. That parasympathetic motor neuron is going to travel down here and cause the smooth muscles of the wall of the rectum to contract to help move the feces down. And at the same time, all right, it's going to cause the internal anal sphincter to start to relax. All right. Meanwhile, up here in the cerebral cortex, you have that conscious sensation like, ooh, I got to go to the bathroom. So you go and sit down, all right, and then you are stimulating the somatic motor neurons. That's going to be voluntary control. Somatic motor neurons only affect one type of effector organ, skeletal muscle. That's it. What's the only skeletal muscle here? Besides the levator ani? The external anal sphincter. You relax that muscle as you're undergoing a Valsalva maneuver, increasing the intra-abdominal pressure, and you push and the feces leave.
So someone's incontinent because they don't have control of that somatic response?
Anywhere in here. There could be several different reasons for that. Somebody that has suffered a spinal cord injury in the lumbar spine, all right, will have this issue because the actual, and we'll talk about it in the urinary system in chapter 24, but it's S2, 3, and 4 are the actual spinal nerves that are part of this. All right, so this is all occurring in the sacral portion of your spinal cord. And so if you're a paraplegic and a quadriplegic, you can lose control of this, and that's one of the possible causes to be incontinent. Yes, and it could be, to answer your question, yes, the somatic motor neuron can be affected, separately, and that would also be a reason for incontinence. Yeah, there's a couple different things. All right, that's not it for the chapter, all right, but that's it for the actual physiology of the digestive system. Now we're going to kind of get into that biochemistry that I wanted to get into. We're going to talk about these macromolecules. All right. So if you've heard the terms like, oh, macro diet or whatever, okay, well, that's what they're referring to are the macromolecules. And so we're going to start off with carbohydrate digestion. All right, where do carbohydrates get digested? Where is this all occurring? And we're going to then talk about proteins, and then we'll talk about fats, all right? So let's start off with the carbohydrates. Quick review, all right? And when we get into the metabolism portion of this course, we'll go into much more detail. So I'm not going to get too terribly detailed here because I could talk about this forever. All right. So chapter 2, we learned, all right, carbohydrates can be monosaccharides, which are what we call simple sugars. Okay, so that's glucose, fructose, and galactose. Then we can have disaccharides, in which we just take two monosaccharides and stick them together. Okay, sucrose, maltose. All right, are some of those examples. Then we have polysaccharides. All right, like starch and cellulose or glycogen, okay, in which we just string a whole bunch of monosaccharides together. All right, they can be the same, they can be different. All right, we'll make what we call a complex carbohydrate. All right, so ideally, the whole point of this, and I hope you understand this concept being in the digestive chapter, is that we're taking stuff that's big and breaking it down into something that's really small. All right, we call that catabolism. All right, we start with the big whatever, and we break it down into smaller whatevers. All right, so that's what we're going to see here. So starch, all right, yes, okay, it's a polysaccharide, so it's pretty big structure. We're ideally going to break it down into individual glucose molecules. All right, that's big, that's huge. Essentially, your body will do that when it's breaking down glycogen. Same, well, not same, but similar. We're going to just pick off individual glucose molecules, and then we can use glucose for ATP production, okay? So when we talk about disaccharides, all right, that's just two monosaccharides. We're just going to break down the disaccharide into each of the individual monosaccharides, okay? So where we're going to see the digestion of carbohydrates are going to be in the mouth and the small intestine, okay? It starts in the mouth. When we start to release, our saliva has that amylase in it. And that amylase, all right, we start to break down starch molecules. So we're going to hopefully start to cleave off some individual glucose molecules, or maybe break some of those starch molecules in half. Anyways, we're going to take something that's big and make it smaller. So we start to break down the bonds between the glucose molecules here. Now, that's good. But when we get to the stomach, when that food bolus gets to the stomach pH unfortunately inactivates, all right, the salivary amylase. So we're going to briefly pause carbohydrate digestion when we get to the stomach. No problem, because we know when we get to the small intestine, it's we're turning the game back on. It's like playing a video game, hitting pause, and then coming back later on, and then pushing play again. That's what we're going to do, all right? We're going to start to I keep wanting to say dehydrate. We're going to digest and break down, all right, those carbohydrates when we get to the small intestine. And the big, all right, enzyme that's going to really start to help further breaking down those carbohydrates is that pancreatic amylase, all right? That's part of that pancreatic juice secretion that occurs, all right, when CCK, all right, starts to stimulate the release and secrete and start to stimulate the release of those enzymes there. All right, so basically, here's what's going to happen. In the small intestine, all right, we are going to make that starch into much shorter strands. It can be individual glucose strands, all right, or maltose, which is two glucose. All right, it's a disaccharide. It's two glucose molecules bonded to one another, all right, or into shorter strands of repeating glucose. All right, because starch is usually just glucose strands strung together. Okay? Questions about that? Because we're going to start to get a little bit more complicated here in a second. All right, so for right now, the only two enzymes that we need to know for carbohydrates is salivary amylase, and it tells you it's in the saliva of the mouth, okay, and it's going to start breaking down starches in the mouth, And then we got pancreatic amylase, which is going to continue the breakdown of those starches, okay, in the small intestine. Okay, because now I'm going to throw at you a couple other enzymes here. All right. All right, so stomach just shot in some chyme into the duodenum here, where we have some of our partially digested starch here. Don't worry about lactose and sucrose. We'll hit that in a second. All right, the pancreas secretes that pancreatic amylase into the small intestine, in the duodenum. It's going to further break down the starch, right, into these long chains of glucose, or into maltose, which are these two glucose molecules attached to one another, or to single glucose. Our ideal goal is to get, all right, all the carbohydrates into these individual monosaccharides. That's what we want, okay? Because then we can absorb them into the cells, all right, and into the blood, and then we can do whatever we want. But we got to get them into these individual, all right, monosaccharides, okay? So the pancreatic amylase is going to help with that. It's going to give us one of these three options, either this chain of glucose, all right, maltose, which is just two glucose attached to each other, or one single individual glucose molecule. See where I'm going with that? Okay, so we need a little help. No problem. Guess what? All right, our small intestine now has what we call brush border enzymes. What's the brush border? That's the microvilli. All right, the microvilli, which sits on the top of our epithelial cells. Okay, so that's what we're talking about. So these enzymes sit right on top of the microvilli here. All right, and there's going to be a whole bunch. We're going to talk about those enzymes now, because again, we've got to get this here, all right, into this here, individual. Okay, so how are we going to do it? Brush border enzymes. Okay, so the brush border enzymes are going to finish the job. So there's two here, all right, dextrinase and glucoamylase. So what will that do? All right, these enzymes, and you can see here on our little chart here, all right, partially digested starch undergoes pancreatic amylase. Here are our three options. We have an oligosaccharide, which is a bunch of glucose molecules strung together. All right, we have a disaccharide, which is maltose, which is two glucose. And then we have one individual glucose molecule. All right, but we want everything to be individual glucose molecules. So we have our dextrinase and our glucoamylase, which will start to snap off glucose molecules off of our oligosaccharide. That's cool. All right, so we're just going to start picking off, bing, bing, bing. All right, so we'll have individual. Then we got maltase. All right, so this is nice because they make it a little bit easy on you. Some of the names of these enzymes will tell you what molecule they're going to work on. Has anyone here ever heard of lactose intolerance? Yeah, okay. All right, and that's lactose is the sugar in milk, okay? So we have an enzyme called lactose, or lactase, excuse me, lactase. That's the enzyme that breaks lactose, which is a disaccharide, right? And lactose is made up of glucose and galactose. And it clips the bond in between, so we have an individual glucose and individual galactose. Well, some of us as we get older, we don't have lactase, so we can't break down the lactose, okay? So here, maltase tells you exactly it's going to break down the maltose disaccharide sugar, all right, into the two glucose molecules. Maltose is just two glucose molecules bonded to each other. Okay? All right, that's not bad. Well, guess what? I got a couple more enzymes to throw at you, but they're easy ones. All right, check it out. Check it out. Okay? When we talk about the digestion of disaccharides, well, what's a disaccharide? Right here. Here are some of our disaccharides. Two monosaccharides bonded to one another. Okay? So when we're dealing with lactose, I told you it's glucose and galactose. So the name of the enzyme, we name it after, except we put ASE on it to tell you that it's an enzyme, we name it after the disaccharide. So you need lactase to break down lactose into glucose and galactose. You need sucrase to break down sucrose, all right, into, again, glucose and fructose. Okay? How do you guys feel about that? Not too bad, right? It's going to get a little bit more complicated when we do proteins. I just want to make it as simple as possible here for you. Okay? So keep that in mind. You've heard of that, lactose intolerance and whatnot. Has anybody ever, I lived with a guy, he was lactose intolerant, so what did we drink? Lactaid? Has anyone ever tried Lactaid? Yeah. It was okay. You know what I really enjoyed? I used to drink shelf milk. Is anyone familiar with shelf milk? Wait. Oh, I can't believe I can't remember the name of it. They call it shelf milk because you can keep it on a shelf at room temperature or even some hotter environments. No, well, maybe it does. I don't know. I bought it in a carton, but once you opened it, then you had to refrigerate it. And a Parmalee. If you go to the baking area, it's something you probably would never buy unless the recipe calls for it. But you'll see it's called Parmalee, and it sits there, and that's shelf milk. But once you open it, you have to refrigerate it. I used to drink that all the time. But almond milk, I used to drink that too. It's just expensive. I don't drink milk.
I drink almond milk because I just get crazy out.
Yeah. Yeah, no, yeah. I mean, I grew up in a house drinking skim milk, which is just like a little bit of milk and a lot of water. You know, I mean, pretty much. So I was used to, yeah, you're right. And I was used to that. So, I mean, going from skim milk to any of these other, well, I can't drink soy, but going to almond milk was no big deal. Plus, I don't have a very good sense of taste anyways. Okay, so now we've got our monosaccharides. That's our goal. We wanted to break down these complex carbohydrates. We have all these enzymes that do it. The brush border enzymes really help with that, okay? Now we're going to finish. Now that we have our monosaccharides, cool. Now we can absorb them right across the plasma membrane of the epithelial cells. And where are they going to go? Right into the blood. Right into the blood. So they go to the blood, they circulate around, they eventually make their way to the liver. All right. Now remember, if we have sucrose and lactose, all right, it involves some other types of monosaccharides, fructose and galactose. Okay. No problem. Guess what? Liver can convert fructose and galactose into glucose. Cells like that. All right. Cells like that. And that's one of the reasons why we don't really consider glucose to be an essential energy or something that's essential that you have to take in through your diet. Because if you're getting just fructose and galactose, no problem. Your liver can convert it right into glucose for your cells. Because remember, the brain loves glucose. That's its favorite. Okay. So what do we do? All right, you get to learn about this in the next section. Not today, but we'll talk about how we make ATP. All right, how we break down glucose, but big part of it is to become ATP. If you're doing good and you're not needing any energy expenditure, but yet you want to save some of that glucose for a rainy day, no problem. We're going to store it. We're going to store it as glycogen, right? Through glycogenesis. Okay, we'll talk about that in the metabolism chapter. Or your body will even convert it to fat. Now, that's not ideal for some folks, especially if they want to keep their body fat composition down. So that's why you hear a lot of people say, hey, don't eat a lot of carbohydrates, because what you don't use, all right, and what doesn't get converted into glycogen, all right, can be converted into fat. So keep your carbohydrate levels down so you can decrease, all right, your fat levels. That's depending on the person. Me, I don't really care what I eat. No, that's a lie. I do. Okay, so cellulose comes from plants. Remember, I don't know if I told you guys this. I teach so many sections, and sometimes I forget what I tell my sections. All right, but if you've ever taken a biology before, when I was in high school, I took just general biology, and we had to learn animal cells versus plant cells. Plant cells have a plant wall. It's literally this wall inside. We have a plasma membrane. Plant cells have a plant wall. Unfortunately, we don't have the enzymes to digest this plant wall. So we refer, all right, to stuff that we can't digest as fiber, all right, which is what we have bulk in our digestive, which is good for actual digestion, believe it or not. You should have, I believe, right now it's 32 grams of fiber a day. It's encouraged, especially if you know anybody that is diabetic, they should be taking tons of fiber, all right, because it helps with controlling their insulin levels, especially during the digestive process, all right? So we don't have the enzymes to break down the plant wall, all right? So that is actually going to be considered fiber, healthy for us for our digestion, and it is encouraged. I encourage everybody to have it. Okay, let's move on to proteins. All right, so proteins, another one of our macromolecules here, okay? So here's the definition of what a protein is, and you need to know it. Because remember, digestion is catabolism. We're starting with something big. We want to make it small, okay? So proteins are polymers, right, which are subunits, right, kind of like how we had the simple sugars or the monosaccharides, all right? Well, it's just we take a bunch of amino acids and we link them together. And what holds each amino acid to the next amino acid is the peptide bond. It's important to know. Okay, so that's essentially what a protein is. All right, so it's a string of amino acids strung together. So guess what we're going to do for digestion? We're going to unstring all those peptides. All those amino acids. We're going to destroy those peptide bonds. That's how we're going to do it. Okay, so that's what happens. We're going to release these individual amino acids. Guess how we're going to do it? Enzymes. Okay, these enzymes are going to target these bonds in between the amino acids here. So here's the thing when we're dealing with the enzymes for proteins. I said it earlier in class. When the cells make them, they got to make them inactivated. Can't have them active, right? Can't be activated until they're outside the cell that makes them. Okay, so that's what we're dealing. So now we got to take another step. Like with the enzymes for carbohydrate digestion, it didn't matter. Okay, they could be active enzymes, so they got excreted out in the lumen, fine. But these, the proteases, which are the names of the enzymes that break down proteins, they have to be inactive. So what we have to do is once they're secreted or excreted out of the cell, we have to take an extra step and activate them. Okay, that's what hydrochloric acid does. Hydrochloric acid does that to pepsinogen. It activates the pepsinogen in the lumen of the stomach, and then when pepsinogen gets converted to pepsin, then it can start activating that active pepsin can then activate pepsinogen into pepsin. All right, so it can go around and helping to do that. So here we are with our pepsin, all right, which is going to be found in the stomach lumen. All right, comes out as pepsinogen. All right, the inactive form gets converted into pepsin. All right, how does it do that? Hydrochloric acid. Hydrochloric acid, which is produced by the parietal cells. What else do the parietal cells secrete out? You remember? Helps with vitamin B12 digestion or absorption in the ileum. Intrinsic factor. Okay, parietal cells, all right, are going to secrete intrinsic factor. Okay, that intrinsic factor is needed for vitamin B12 absorption in the ileum because what do we need vitamin B12 for? Right, normal erythrocyte production. Okay, want to have normal red blood cells, need to have that vitamin B12. Okay, so stomach's low pH will help to activate the pepsin from the pepsinogen. All right, and all right, proteins can be big. Remember, I told you the minimum they have to be 200 amino acids long. That's long. All right, so we can start to physically break down, all right, those proteins just with hydrochloric acid because it causes denaturation of the proteins. Remember, you heard me talking about it when we talked about the buffering system in the blood. Our pH in our blood has to be that 7.4, all right, because if it gets too acidic, if it gets down to 7, 2, 7, all right, 6, 9, 6, 8, God forbid, then our proteins are just falling apart. They fall apart, okay? And I'm going to review with you when we do metabolism all the functions of proteins. All right. So we saw what happens in the stomach. Now let's move into the small intestine. All right. This is where we're going to start to further the breakdown of proteins. So we have 3 enzymes that are made in the pancreas and secreted in the pancreas. Notice their names. It's got GEN, all right, on the first 2. That's the inactive form. Okay. And then this pro-carboxypeptidase, that's a mouthful. All right, that's also inactivated. They're all inactivated. Okay, so it's cool because this first one, okay, trypsinogen, when it gets activated by enteropeptidase, that's this enzyme that sits out in the small intestine. Because remember, we got to activate it. It activates trypsin, and then it activates trypsinogen into trypsin. And then Trypsin then will go and activate this one, it'll go and activate this one, it'll go and activate this. All right? So it's like a nice cascade effect here. We just have to start off by activating trypsinogen into trypsin. All right? Trypsinogen is what we call a zymogen. Okay? Questions about that so far? All right. So you can see once we've activated, all right, all our inactivated enzymes, now we can get down to the nitty-gritty. Now we can start to break down, all right, these proteins, these denatured proteins, all right, into smaller structures. Because remember, we want to get down to the individual building blocks. In this case, that's amino acids. So these first two, trypsin and chymotrypsin, are going to break bonds in between specific amino acids. It's not just willy-nilly. It's not just here, not just there. All right. So what that'll do is that'll create, all right, strands of peptides. What are peptides? Peptides are just small strands of amino acids strung together. If there's just two, we call it a dipeptide. Okay, 3, 4, 5, 6. All right. It makes just smaller peptides. Okay, so we're getting close. We're getting down to those individual amino acids. We're just not there yet. But these guys help to take maybe, you know, a 20 or an 18 long strand amino acid, and then it just starts to cleave off like and make these smaller peptides, which are maybe three amino acids long or 4 amino acids long. All right, helps us out with that. Okay, this last one here, carboxypeptidase. All right, that's going to cleave the amino acid off of at the carboxyl end. Now, I know I haven't gone over this with you, and I'll do it when we do the metabolism. You have 2 ends to an amino acid. At one end, it's the more acidic end. All right, that's the carboxyl group end. And then at the other end, you have what's called an amine end. All right, that has the nitrogen bound to it with some hydrogens coming off of it. Okay, so this enzyme is going to cleave the amino acid at the carboxyl end, okay, which I'll show you on this picture. I'll come right back to this. All right, so here is our peptide, okay, and so you can see trypsin and chymotrypsin, they'll go along and break the peptide bonds at specific amino acids inside of our denatured protein or our oligopeptide. But you notice carboxypeptidase, here's the carboxyl group here. The amine group is down at this end here. All right, it's going to specifically, all right, break the bond here between the carboxyl group and this amino acid here. Okay, so this one is our specific right here for this one end. All right, whereas trypsin and chymotrypsin are still amino acid peptide bond specific. All right, but this guy here is going to be down here by the carboxyl group. All right, I think that's everything I wanted to cover on here. Yep. Okay. And then we have these 2 final, all right, amino acids, not amino acids, I keep on saying amino acids, brush border enzymes, all right, which is called dipeptidase. Obviously, when you have 2 amino acids, all right, bound to one another, that's a dipeptide. So this, all right, enzyme will cleave here, giving us, all right, 2 individual amino acids, and then you have this enzyme here, which is an aminopeptidase. If you have a peptide that is, I don't know, 4 amino acids long, it just starts to cleave off, all right, individual amino acids. Okay. So again, our goal is to get down to the building blocks of proteins, which are these individual amino acids. So what happens? Now the epithelial cells can absorb those amino acids into the cell, and that cell can then use those amino acids to maybe make some proteins that it needs. And if it doesn't, no problem. It can then package it up if it wants to make hormones with it, if it wants to send it into the bloodstream so it can travel throughout the body to other cells that need it, okay? Or in some situations, we can convert, all right, those individual amino acids into glucose through the process of gluconeogenesis, but they have to go to the liver to do that. All right, the only organ in the body that can perform gluconeogenesis is the liver, and that's making of glucose from non-carbohydrate sources. Okay, so that's what this picture here is showing us. All right, here we're getting our denatured. Okay, we're getting our denatured protein here into the duodenum here. All right. And so we have, all right, the pancreas releasing, all right, the inactivated, all right, proteases that get activated through enteropeptidase, all right, which is here in the small intestine, converts, all right, trypsinogen into trypsin, which will then auto-activate other trypsinogens into trypsin, but trypsin will also activate, all right, chymotrypsin and then our carboxypeptidase to start to make, all right, these larger oligopeptides into smaller peptides. And then our brush border enzymes here, all right, dipeptidase and aminopeptidase, will start to clip off, all right, individual amino acids or break the bonds in between these two dipeptides. Result, individual amino acids absorbed into the cell. The cell will use what it needs. Whatever it doesn't need, it ships off into the blood, travels throughout the body to be used for whatever. Cool? Questions? Almost there. All right, lipids. Okay? Keep in mind, when we're talking about lipids, right, they are not water soluble. Okay? They're water insoluble, just like oil. All right? You pour it into water, they don't mix. Okay? So they're not water soluble, they're lipid soluble, obviously. So if you remember the classification of lipids, we have triglycerides, all right, phospholipids, steroids, and eicosanoids. Okay? So we're going to talk about triglycerides and cholesterol. Okay? So if you remember, triglycerides are made up of a glycerol group and 3 fatty acids. Now those fatty acids can vary. They can be long, they can be short, they can be medium. So we call them, if they're long, we call them long chain fatty acids. The longer they are, the more energy you can get out of them when you're starting to break them down for energy. Okay? So that undergoes a process called beta oxidation here. All right, so again, we'll need some enzymes. We'll talk about those in a second to break the bonds, all right, between these structures. Cholesterol, I love this about cholesterol. Guess what? It doesn't need to be broken down. Okay? It doesn't need to be. Because the cells, if they need to, they can incorporate cholesterol right into the plasma membrane. All right, we're going to use cholesterol for a lot of things. We want to make steroids, we need cholesterol. All right? You want to make some hormones, you need cholesterol. Okay? So it's very helpful. So there's a lot of things that we can do with cholesterol. All right? So that's why I'm not so upset unless your cholesterol levels are off the charts. You got 300, you know, your cholesterol levels are like 300. You know, that's bad. But I do encourage people to take a cholesterol into their diet naturally. It's not necessarily a bad thing. All right. So we start the breakdown, all right, of lipids, not really the breakdown, all right, but the lingual lipase, which is found in our saliva, okay, when it coats the bolus there, once it enters into the stomach, then it's activated, along with gastric lipase. All right, it's got gastric lipase, the lingual lipase starts to break down these fats. I'll talk about that later. All right. So, for the most part, remember, one of the functions of fat is cushioning, insulation, but one of the ones I haven't really talked about so much is energy. All right. It is a great way to have energy, and one of the best energy sources from our lipids are triglycerides. So I'm going to draw a really horrible picture. Here's the glycerol group. All right, and then you got your 3 fatty acids attached to it. So here in the stomach, what we're going to do is, all right, we're going to just clip off one of these fatty acid groups. All right, so we're going to go from a triglyceride to a diglyceride. So you'll have the glycerol group with 2 fatty acids and then 1 individual fatty acid by itself. Okay, so that 1 individual fatty acid, Okay, no problem. We can do a lot with that. We can actually start breaking that down. We can absorb that in. All right, we can move that into the small intestine. There's a lot to be done here, okay? So all this occurs without the need for bile salts. We haven't started that emulsification process, all right? Now, we exit out of the stomach and we go into, you guessed it, the small intestine. So now CCK starts pumping the bile salt, the bile into the small intestine. The pancreas starts pouring, all right, the pancreatic enzymes into the small intestine. So we have this pancreatic lipase, all right? And that pancreatic lipase is now going to any triglycerides that are present. It's going to convert those triglycerides now into monoglyceride, meaning you have the glycerol group with one fatty acid attached to it, and then we have two individual fatty acids all by themselves. Okay, that's cool. All right, so we're starting to get there. We're breaking stuff down here. So now what we're going to see here, all right, due to bile, we're going to have the emulsification process occurring. All right, that's when we take a large lipid droplet and turn it into a smaller one. All right, so we call that process emulsification. All right, that's what bile salts really do. Now I can go into more detail, but I'm not, because I know when we do the metabolism, we will. Okay, so I'm just kind of setting the groundwork here, doing the foundation. Okay, so in order to undergo emulsification, we need bile because we need the bile salts in there. And basically, what this will do is, all right, we'll get these small molecules, all right, these bile salts will surround. This term here, amphipathic, means you've got a polar end to this molecule and a non-polar head. We saw that with phospholipids. Remember the phospholipids in the plasma membrane? You had your polar head with the two non-polar tails. Okay? If it's polar head, it's water-loving, hydrophilic. All right? The non-polar end is hydrophobic. Okay? So what we're going to see here is something similar. All right, we got our molecule, all right, that's going to have a polar head and a non-polar tail, so it'll look kind of like this, just one individual. All right, and what it'll do is, here's our fat droplet, okay, and these polar heads will surround it. All right. Hold on, I'm drawing it wrong. Actually, why am I doing this? I do this every class. Let me show you a way better picture. Instead of my horrible. All right, here you're seeing, this is what happens. So here's our lipid droplet here, all right, and then these bile salts, all right, with their polar heads and their non-polar tails, all right, will line up and surround, all right, that lipid droplet and form what we call a mycel. Okay, so you got the lipid in the middle, and then you've got the bile salts surrounding it here. Okay, and this helps to emulsify. And I'm going to tell you, one of the things that occur, let me make sure. Oh no, I already went over that one. Hold on. I'm going to talk while you write. Okay. So what does this do? Okay, this actually allows, all right, this process is going to allow, all right, that enzyme, all right, that pancreatic lipase, more access, all right, to the triglyceride to help to break it down. All right, it's going to help break it down, all right, into smaller. That's the whole point of emulsification. We start with a big lipid droplet, and now we're going to turn it into a smaller lipid droplet so we can get it closer to absorption here. Okay, so what will happen is it breaks it down, all right, and then the cell, I'm jumping ahead here, but the cell will, the epithelial cells will absorb, all right, these small lipid droplets, and the bile salts don't go into the cell. They stay in the intestine, where then they can be absorbed and then sent back and be recycled to be used again. Very small amount of the bile salts actually get excreted in the digestive tract, usually about 10%. They actually have drugs. I'm going to save that. I'm going to keep jumping ahead. Sorry. I want to just finish this chapter up. All right. So keep in mind, all right, when we form these mycels, okay, we're going to have the heads, which are polar, they are hydrophilic, so water-loving. So the heads will be towards the aqueous fluid, which is the majority of it is water. Okay, so it's not a problem, it's good. All right, so what we'll see then, all right, is that these lipids move towards the simple columnar epithelial cells, all right, enter into the cell. All right, the bile salts stay outside. They stay in the lumen, which they'll get hopefully reabsorbed. 90% of them do. 10% get excreted. All right, we're going to go into this part in much more detail, so I'm going to kind of run through it fast. But basically, inside the epithelial cells, what will happen is we are going to form these structures called chylomicrons. All right, basically, that is just a lipid that gets surrounded by proteins. You've heard of LDLs and HDLs, good cholesterol, bad cholesterol. All right, and VLDLs, very low density lipoproteins. That's what these structures are. They're chylomicrons. So now we basically gave up one wrapping for another. We had a wrapping outside in the lumen from the bile salts. Now inside the cell, we're going to wrap up these lipids with a protein. Okay? And so these proteins, these lipids that are wrapped in proteins, we call them chylomicrons. So what's going to happen? All right, the cells will then get rid of these chylomicrons, all right, through the process of exocytosis, all right, and they'll go into our lacteals, because remember, all right, the lacteals are those small lymphatic vessels, all right, that are going to drain the digestive system and then drain into the lymphatic system, and eventually the lymphatic system will dump, all right, those fatty droplets, these chylomicrons, all right, into venous circulation. All right. Eventually, this will all make it to the liver because that's what we want. We want these lipids to end up at the liver, all right, because we'll talk about that, what's going to occur at the liver, because we're going to convert these lipids into something else. We want to make cholesterol, or if we want to make some other structures and some other tissues that might require some of the fats. We'll talk about that, all right? I'll get into that in much more detail here. I just want to finish this up here. All right, so that's what we're seeing here, okay? So our fatty droplets come in, okay? They undergo the emulsification process. All right, we break them down into smaller lipid droplets. They're surrounded by these myceles, okay? Which makes it more appetizing, so to speak, to the pancreatic lipase. Right, that helps to break them down into their smaller molecules of fatty acids, monoglycerides, and whatnot. Okay, they enter into the cell, all right, where they get packaged up in a different way, all right, with proteins, and then they'll get excreted into the lymphatic system and eventually make their way to general circulation, to the liver, which we'll talk about when we do the metabolism. All right, so let's finish up here when we talk about the other structure or other items that actually, we talked about lipids, we talked about fats and carbohydrates. What about the other stuff that you take in? All right, water, all right, like I said before in the beginning of class, most of the water is going to be absorbed, all right, in the small intestine, okay? That's a lot, 8 out of 9 liters, the majority of it, okay? You would think that the large intestine would do it, all right? But no, the small intestine. The large intestine does, all right, for the most part, right? Eventually, though, we're going to see here, all right, that the water will move into the blood circulation through osmosis, passive process, regarding the movement of water from an area of high concentration to an area of low concentration through a semi-permeable membrane, okay? Electrolyte absorption, mainly in the small intestine. The large intestine does also, but we'll see here with iron, all right, it has a regulated amount of absorption going on depending on levels of iron in circulation and in your body, all right? And so we have this hormone, okay, hepcidin here, which is going to come from the liver. So if there's high levels of iron, okay, in the body, then this level of hepcidin will increase because it inhibits the absorption of iron. Okay, if there's low levels, we'll have low levels of hepcidin. All right, and finally, vitamin or vitamin absorption. Okay, depending on if it's water soluble or fat soluble. All right, we'll see with our fat soluble, the A, D, E, and K. All right, which are going to occur in the small intestine when we'll see lipids, all right, with the micelles being absorbed, are water soluble, are going to be absorbed throughout, all right, because they're water soluble, they can diffuse and absorb into the water, all right, so that can occur anywhere along, all right, the digestive tract here. We'll see quite a bit of it, obviously, in the small intestine, some of it in the large intestine here, all right. Keep in mind, B12 here, we need that for our production of our red blood cells, normal red blood cells, but because of it's so large, you probably don't remember receptor-mediated endocytosis. All right, this is the third type of active process that occurs, okay? And so it's how a cell takes something from the external environment to the internal environment. Basically what will happen is vitamin B12 will be absorbed through like a interstitial fluid water droplet kind of formation. But when it enters into the cell, all right, the cell puts what we call a clathrin coating. It's a specialized protein coating that gets put around that vesicle, right, that has the vitamin B12 inside because of its size, and that helps to identify, all right, that the inside of whatever is in this envelope here, all right, it helps to identify to the cell what that is, okay, and what it can be used for, okay, and then intrinsic factor, all right, we need that. That's going to be absorbed in the ileum, okay, and that's going to help the increased absorption of vitamin B12. All right, I think that is it for this part. How much time do I have left? Fine. We will not complete. We'll talk about this. Okay, this is what you got to do for me. All right, I know we're going on break and whatnot. Let me stop recording.